therefore an exciting opportunity to introduce our speaker because she is a member of our own department here. Dr. Jorgensen actually is a postdoctoral fellow here and works in Dr. Bigham's lab. She is a population geneticist. She got her PhD from Wayne State University and her master's from University of Edinburgh. And throughout her academic career, and here alongside all of us, she's studying how human environmental interactions affect genomic and genetic variation in human populations. So her work is very exciting in bridging the genotype, phenotype areas, and she uses a multidisciplinary approach, which I'm sure she will share with us today. So without further ado, let's give a round of applause for our very own Dr. Jorgensen.
There's not like a little dongle or anything, right? Okay. I'll do this. Okay. Thank you all for coming. We're kind of a intimate group, so if you feel the need to interrupt me and ask questions, like we can make this more back and forth or conversational. This is kind of, I guess, my swan song in the department. I've been here for 2 years and my contract is ending next month. And Abby was kind enough to invite me to give a Beck to highlight some of the projects we've done together and some of the projects that I'm hoping to continue in the future. So looking forward to some feedback, some thoughts, critiques, whatever you have to say. And a lot of my research focuses within this underlying question of how do humans evolve in response to their environments, and in turn, how do the environments shape the genetics of humans? So humans have settled some of the most challenging environments on Earth, whether that's extreme high altitude, extreme cold, or extreme heat, and in turn, these environments have shaped our genomes over the years. So some really classic examples of that is lactose tolerance. Another one is the shift to crop domestication and sickle cell mutation in response to malaria. And I'm particularly interested in situating this research within evolutionary anthropology because I see it as these genetic traits under selection are manifesting themselves physiologically, and it's within this framework that we can really build a dialogue for how these underlying pathways can connect genotype to phenotype, and we can communicate to more clinical outcomes and eventually better healthcare by placing this context of evolutionary history within our understanding of social processes and context. And what I touched upon is I answer a lot of my research using both genetic data but also phenotype data, and I'm really interested in uncovering the mechanism of how these biological processes actually manifest themselves. And it's within this that we can really uncover how this past evolutionary adaptation can continue to shape health in present-day human populations. And I was really drawn to using population genetics as a tool for understanding this genotype to phenotype pathway because I saw that there was really a lack of diversity in current population genetic studies. So this figure on your left is actually from yesterday, and it represents the total catalog of genome-wide association studies related to health. And as you can see, there's really a dearth of research in any population that's not Northern European. So I'm really interested in how we can use genomic research as a tool to better identify and characterize this diversity because it can really affect how healthcare works in the future. And I put up a few examples there. So if you're talking about precision medicine, you really can't identify genomic variants that could affect the metabolism of medicines or what you might need to identify and diagnoses of disease if you haven't sequenced any part of the population. And a lot of this knowledge is really used to inform public policy in the future. And you can't inform public policy in the future if you have no knowledge related to these populations. And so much of my research focuses on working with understudied populations to really address this lack of diversity, but also so that we can shape the way that biological research is conducted. As anthropologists, it's really important to me that all of these research projects are really community-based research projects. So what this means to me is I'm committed to capacity building in different senses. And I work, as you'll see, in a few different field sites, but in Nepal in particular, they're really interested in scaling up their research from looking at single variants in BRCA1 and BRCA2 to whole exome or whole genome sequencing to really get into that space of precision medicine. So capacity building at that field site in particular is transmitting and training local scientists there in order to conduct the bioinformatics research on their own. And what this can mean at different field sites is writing into grants to donate the actual equipment that they need to carry out this research on site in hospitals. And so I'm going to be talking about 3 different projects that I have going on that really focus on connecting this genotype to phenotype and showing how our evolutionary past can still be reflected in modern genetic variation. And one of these projects looks at high altitude adaptation. I'll be talking about it in the Andes. And another one looks at dietary adaptation in the Andes, as well as more broadly in the Americas. And the last project is multispecies genetics, and looking at how the environment and our cultural behaviors related to diet can really come together and affect other organisms and can really inform co-evolutionary relationships. Okay, so for high altitude adaptation, this is a project that is near and dear to Abby's heart, and when she asked me to do Beck, I was like, I don't know if I can talk about high altitude adaptation to your face in your department. And she's like, don't worry, I won't be there. So hopefully she won't watch this recording later. But if you're not aware, so despite many environmental challenges in the Himalayas and the Andes, such as cold temperatures and lower oxygen conditions like hypoxia, early human populations actually moved into these areas thousands of years ago. So in the Andes in particular, they moved to that area about 12 to 9,000 years ago, depending on what archaeologists you talk to. And it was even earlier for the Tibetan Plateau, as early as 30,000 years ago. And successful adaptation to these different high altitude environments would have required both cultural and biological adaptations. And among these, one of the most studied in anthropology has been the genetic component to high altitude adaptation or hypoxia. And there's a lot of different genes involved in oxygen sensing and metabolism. And here I want to highlight the hypoxia-inducible factor pathway, or HIF pathway for short, because it really mediates the majority of the cellular response to lower oxygen conditions. And there's been many previous studies that have actually identified several genes within this pathway under positive selection in relation to high altitude populations. And the box I've drawn here is to point out 2 genes in particular, EPAS1 and EGLIN1, that have really consistently emerged as being under positive selection in both of these populations. So in Tibetans in particular, variants in these genes have been associated with a lower hemoglobin concentration. And for Andeans, variants in eglin-1 have been associated with a higher aerobic capacity or elevated VO2 max. But despite many rich studies in this region, particularly by Abby, the actual precise genetic variants that are causal for mediating this response to lower oxygen conditions have still not been identified in these populations. But what I want to point out in this figure is that the majority of genes are under selection in the HIF pathway, and the colors here represent genes that are under selection in multiple populations. So what this means is that natural selection hasn't acted on the HIF pathway as a whole, but rather on very selected targets within it in different populations. And one of the reasons that I'm really interested in looking at this question in high altitude populations is we're working with collaborators at the University of Pennsylvania School of Medicine, and we really want to leverage sort of this natural experimental laboratory of populations living at high altitude for thousands of years and having variants that are adapted to these conditions and identifying them and then using them as future targets for reversing microhypoxic environments of tumors. So if we can identify these variants that are mediating this response along the HIF pathway, perhaps they could be useful in other more direct clinical contexts. And one of the reasons that it's actually possible to do a lot of this research is the cost of whole genome sequencing has dramatically decreased in the last 20 years. So it's gone from $100 million per genome about 23 years ago to the quote we got the other day was like $150 per genome, so pretty great. And especially when you combine whole genome sequencing with other physiological and phenotype data, you can really get at what are the variants that are mediating this physiological manifestation. And particularly for high altitude, one of the patterns that I noted before is that these populations have unique phenotype adaptations. So Tibetans have that lower hemoglobin concentration, and Andeans have that elevated aerobic capacity. But despite Andeans not having that lower hemoglobin concentration, they're still protected against pulmonary hypertension, chronic pulmonary hypertension compared to low altitude populations that go up to high altitude and those that are not evolutionary adapted and live at high altitude for chronic periods of time. So they don't have this adaptation, but they're still protected against that condition. So something that I set out to investigate was whether there was something unique about the cardiovascular system of Andeans in particular that's providing protection against pulmonary artery pressure. And my hypothesis in particular was, is there a genetic component to protection against pulmonary hypertension in Andeans? So to answer this, I used a combination of different types of data, whole genome sequencing and genome-wide array data, as well as some physiological measurements such as hemoglobin and VO2 max, as well as RNA-seq. And this data was collected before my time in the lab, but I was lucky enough to join Abby's lab and get to process and QC all the data. So very intimate knowledge of the whole genome sequencing data in particular. So for this, we used a high altitude population recruited from Cerro de Pasco, Peru, as well as a lowland control that was recruited from Palenque, Mexico, Chiapas. And for looking at these selection scan analyses in particular, I used a variety of different statistical tests to detect whether there was any genetic component under selection that could be linked to protection against pulmonary hypertension. And what I would really expect to see in these statistical tests for any mutation that was beneficial is that it would rise in frequency in a given population over several generations. So a de novo mutation would occur, it would be selected for, and then as you can see over time, it would rise to quite a high frequency. And what that would translate to in the actual statistical tests is we would see this site in the genome that has preferentially increased in allele frequency compared to other surrounding regions of the genome. So it would result in this really nice peak that you see there. And the example that I'm showing is actually lactase persistence or selection for drinking milk past infancy in Europeans. So I used several different types of statistics in order to look at positive selection. Some of these look at single alleles or single nucleotide polymorphisms, SNPs, and some look at haplotypes, which are groups of the genome that are inherited together from a parent. And the reason that I apply all these different types of statistical tests is I really want an orthogonal positive result that is not a false positive. I don't want something that is more reflective of a demographic event, such as a population bottleneck, and I want to make sure it's a true sign of positive selection in the population. So after completing several of these selection scan statistics, I found evidence for positive selection within this gene, EPAS1, which was located within the HIF pathway in Andeans. And this is really exciting because, as I mentioned before, there were variants within EGLIN1 associated with a positive or a beneficial adaptation in Andeans, but this was the first study to date that has found any signature of positive selection in EPAS1 with Andeans, which was only previously associated with lower hemoglobin in Tibetans. And when I looked at this gene more closely, I found that there was a single SNP that was unique to Andeans. So it was at about a 7% frequency in the population, but totally absent in every other global population. And it produced a missense mutation in the protein. So for the other phenotype associations, I decided to look at this SNP in particular. And first, I looked to see if there was an association with either hemoglobin concentration or exhaled nitric oxide. And I looked at this in our larger Andean cohort of genome-wide array data. And I found that while there was no significant association with hemoglobin, we did have a significant association with exhaled nitric oxide. And this can help mediate the body's response to hypoxia because it improves vasodilation or increased blood flow, and it could potentially modulate pulmonary vasculature and help with regulating pulmonary artery pressure, reducing the chances of pulmonary hypertension. And next, we looked at the functional effect of this SNP in vivo, and we worked with our collaborators at University of Pennsylvania to generate a knock-in mouse with this SNP using CRISPR. And after we exposed both the mutant mice and the control mice to a series of different hypoxic conditions, we again found no significant association with hemoglobin. And then, okay, so no significant association with hemoglobin. We next looked at the different effects of these hypoxic conditions through two different tests, mean right ventricular pressure and the Fulton index. And what we found is that for both mean right ventricular pressure and the Fulton index, they were significantly decreased in our mutant mice compared to control mice. And what this means is that our mutant mice had less inflammation associated with pulmonary hypertension in hypoxic conditions. And then lastly, when I looked at overall transcriptional activity of these mice, I found that this mutant allele actually results in decreased transcriptional activity, and a gain of function allele stabilizes this EPAS1 protein, which led to pulmonary hypertension under normoxia. So putting all of these pieces of evidence together, I found that mice with this mutation had less inflammation associated with pulmonary hypertension and hypoxia, and that this overall mice with this mutation had decreased hypoxia-induced pulmonary hypertension due to overall lower transcriptional activity compared to control mice. And in our Andean Quechua cohort, I found positive selection in EPAS1 as a whole with our whole genome data, as well as this SNP in particular being significantly associated with exhaled nitric oxide. And what all this means together is that EPAS1 for the first time could be associated with a altitude adaptive phenotype in Andeans. So in Tibetans, we know it's associated with lower hemoglobin. But in Andeans, it could be associated with differences in their actual pulmonary vasculature, which could lead to decreased chances of pulmonary hypertension in hypoxic conditions. And this project, I think, is really interesting because there's not a lot of studies to date that have collected both whole genome data and phenotype data from human populations in the field. And I think that's really the difference that anthropology can make to these types of studies when we're looking at what are the long-term evolutionary outcomes of how our genomes are really shaped by all these environmental processes in the past. And I was lucky enough to receive funding from a few different sources to expand this project. So if you've hopefully chatted with Abby or myself in the past, we've been going to Nepal for the last couple years to collect similar amounts of data. So looking at whole genome sequencing and collecting RNA sequencing and other physiological markers with our Sherpa participants. And we hope to have some really interesting data from that soon, but it was a really great project, so wish I could show more pictures. Okay, and so while this genetic component to lower oxygen in the mountains in high altitude conditions is a really important part of why populations were able to settle in these regions, I'm interested in another reason why human populations were able to successfully settle in the Andes thousands of years ago. And I believe it was largely due to the availability of a very plentiful food source, the potato, if that was not obvious. So just for a bit of background, once humans moved into the Andes about 9,000 to 12,000 years ago, they actually began domesticating the potato relatively rapidly. So it was domesticated anywhere from 5,000 to 10,000 years ago in southern Peru, right around the Lake Titicaca Basin, which is the largest freshwater lake in South America. And the potato is a really calorie-dense food source. It does great in harsh, high-altitude conditions and in cold temperatures. And it's possible that if ancient Peruvian populations relied on the potato to survive and began consuming potato starch in greater quantities, as well as kind of this general shift to crop domestication, it's possible that this could have led to a genetic adaptation to better digest potato starch more efficiently, and we could still possibly detect this adaptation in present-day Peruvians. So the question I set out to answer was, can I detect adaptation to digesting starch, specifically potato starch? Was this an important food source that would have been reflected in their genomes? And I applied many of the same statistical tests that I described previously, but this time I did it through a targeted genomic approach. So I analyzed in particular these 4 gene regions that were associated with starch digestion. And just to give you a very brief overview, for starch digestion, when you eat a starchy food, the saliva, the enzymes in the saliva in your mouth break down the starch polysaccharides into smaller disaccharides. These can contain things like maltose and sucrose. And then this passes to your upper small intestine, where enzymes secreted from your pancreas continue to break down these disaccharides. And then the final step of starch digestion actually also occurs in your small intestine. And it's the most important stage of starch digestion because it's at this step that both the majority of glucose is both produced and absorbed. And glucose is the most important source of energy in all organisms. So I thought, if I'm going to find any signature of positive selection within these genes, it's most likely going to be either in MAGAM or SI because it's associated with this really important step of glucose production. And I knew I was on the right track because in 2018, a paper came out by John Lindo and all, and they found signatures of positive selection in MAGAM, but in ancient Peruvian individuals. So I used publicly available data from the 1,000 Genomes Project looking at Peruvians, Han Chinese, and West Africans using much of the same statistics as the previous project. And for these selection scan statistics that require control populations, I chose West African and Han Chinese because, relatively speaking, they've had a shorter history of potato starch consumption, less than 500 years, and they consume less potatoes per capita. But it is really, really hard to find a population that doesn't eat potatoes today. So, caveat of research. And after applying many of these statistics, I did not find any significant results in the earlier steps of starch digestion in AMY1 or AMY2, but I did find, as you can see, a cluster of significant SNPs within the MAGAM gene. And very excitingly, when I looked at the results across my other statistics, I found that this exact same region was under selection within MAGAM. And these convergent results together really suggest that there's a unique haplotype in Peruvians associated with starch digestion in some way. But just to be sure, I also ran the same statistics in my control populations and found that this haplotype was not present or under selection in either West Africans or Han Chinese. So this haplotype in MAGAM is under positive selection. It's associated with starch in some way, the process of starch digestion, but I was really interested in whether it could be linked to potatoes in particular. So I ran this statistic EHH to estimate an age for when this haplotype would have arisen in the population. And it assesses the age of the haplotype using linear regression. So you pick your most significant SNP and then it calculates core decay over generation time from that site. And the result was it estimated an age of about 382 generations, or approximately this haplotype is about 9,547 years old. So when I plot that on my timeline of when ancient Peruvian populations moved into the Andes and roughly when they began domesticating potatoes, it very much lines up with the earlier estimation of when potato domestication would have occurred. And this was really interesting and continues to be a point of conversation with many archaeologists because, as you notice, it's leaning much more toward the molecular estimation of potato domestication and is earlier than the archaeological estimation of potato domestication. So this could be just a lack of preservation in the archaeological record, or it could indicate that people were in the Andes and started this cultivation domestication process much earlier than what archaeologists estimate. So, ongoing conversation, as I said. And looking at this haplotype in particular, it lies within a region of particularly high transcriptional activity. It's within this region called intron 34. And intron 34 is associated with the rest protein. And the rest protein does exactly what it sounds like it does. It actually suppresses transcriptional activity, but we don't know the exact function of intron 34 at this time. So it's possible that this haplotype under selection in Peruvians is manipulating the transcriptional ability of the rest protein. So it could either be suppressing it further or stopping it from suppressing and allowing increased expression to happen. And putting this in a broader context is this altered gene expression of magum could have led to Peruvian populations more efficiently breaking down potato starch carbohydrates into sugar, leading to enhanced glucose production or more energy. And for early populations moving into this environment, having this unique genetic adaptation to extract greater nutritional value from this food source could have been a benefit to ultimately surviving in this environment. But as you may recognize, our diets have drastically shifted from what they once were 10,000 years ago, and we're an agricultural-based society today. Many of these populations have been influenced by a westernized diet, so higher processed fats and sugar, and up to 55% of our caloric intake is starches today. So what could have been an adaptive trait to extract greater nutritional value from a starchy food source could be causing the body to digest greater amounts of carbs, producing more blood sugar that enters the bloodstream. And if this is beyond what the insulin produced by your pancreas can help the body absorb, this can lead to, over time, chronic insulin resistance. So again, over time, this chronic insulin resistance could lead to other future negative health outcomes such as prediabetes or type 2 diabetes. And when we look at populations with Hispanic or Latinx ancestry, they are in particular associated with higher risk of glycemic and other metabolic related health issues such as prediabetes and type 2 diabetes. So with this context in mind, I looked once more at this haplotype in my Peruvians, this time using an algorithm called RFMix2. And what this basically does is I assemble a reference data set, and then I train the model to look at my target population and assign different ancestral segments to my ancestral population from this reference data set. So what I found, looking at the magum region in particular in Peruvians, is that it's overwhelmingly of indigenous American ancestry more broadly. And what this could mean is that other indigenous American populations beyond Peruvians could possess similarly unique genetic variants to digesting starch, whether that's potatoes or a different food source. And when you shift that genetic adaptation to a modern context that could be impacting diet and health today. So something that I'm expanding hopefully next year is moving this project to recruitment in Guatemala, which is a population with a long history of starch consumption, but in this case, maize domestication and consumption. And we've already set up a field site in Guatemala City with my collaborators at Santa Cruz, and hopefully we're gonna start recruitment of whole genome and looking at some physiological markers related to glucose there. And again, I just want to say that something that's really important to me for all these projects is that this has some kind of benefit to the community. So these are really important health crises in both Peru and Guatemala, and particularly is drastically underfunded and understudied in indigenous American populations with the government. So it would be a goal of this project to hopefully examine the interplay of genetic variation within these populations and how we can use it to inform healthcare practices or dietary management or more long-term community public policy related to diet and health. Okay, so lastly, I'll be flipping this lens a little bit and talking about how we can use a multi-species perspective to look at this human-potato relationship and how we can untangle patterns of human migration using that co-evolutionary relationship. So if we go back to this timeline of when potatoes were first domesticated in southern Peru in the Andes about 10,000 years ago, we know that humans had this plentiful, hardy food source. It was domesticated in this region, and they would have spread to new regions from that area carrying this food source, but we don't know what directions they spread in, and we don't know the timing of when they would have migrated and habitated new regions of the Andes. And I'm using a co-evolutionary model to approach this question of looking at migration because more traditional lines of evidence are not available. So usually you would look at ancient human DNA to get at these questions of human demographic processes, but in this case in the Andes, there's only about 6 ancient individuals that have been identified to date that predate 5,000 years, and they're roughly from the same 2 sites. So you're not going to be able to answer a lot of these broader migrational questions with that. And you might be thinking, okay, well, why didn't you just sequence the potato if you want to see how people moved with the potato to new regions? Well, because the potato was domesticated in Peru, there's actually over 4,000 strains of Solanum tuberosum just in the Andes today. So logistically was difficult to sequence every one of these varieties to reconstruct their relationship to each other. So instead, I used species of the Andean potato weevil. So there's approximately 10 species, and this insect in particular is the most severe and widespread potato pest in the Andes. And it has really interesting, unique biological restrictions, which would have made independent speciation impossible. So it's only through human facilitation that this insect was able to spread and diverge throughout the Andes. So some of these biological restrictions are it's completely flightless, it has no wings. In different experiments, it can't walk more than 500 meters on its own. It can only live in the Andes. It's completely cold adapted. So if you bring it down to warmer temperatures, either in the Amazon or along the coast, it has reduced fecundity and can't consume its food or reproduce and survive. It also can't climb over any walls greater than 1 meter high. And it can't cross running water. And if you've ever been to South America, there are lots of little streams that dot the landscape. And most importantly, this insect complex is completely dependent on the potato for both its survival and reproduction. So what happens is, at the larva stage, it'll burrow into the potato, it'll feed on the potato from within, it'll complete its growth cycle, and then as an adult, it'll emerge from the potato, and it'll burrow in the soil next to the plant and feed on the leaves. So you get these characteristic half-moon divots, and it'll keep feeding on the potato sometimes. But what's really interesting is that when it's burrowed in the potato at the larval stage and feeding on the potato, you actually can't tell externally that there's a pest inside the potato. So it's only when you cut it open and you go to eat it or something that you realize, oh, there's a charming Andean potato weevil who's eaten half of my potato from the inside. So despite all of these biological restrictions, like I said, this insect complex is the most severe and widespread throughout the Andes, and it really could only have been possible as a consequence of human movement. So I hypothesized that as people carried their potatoes to new regions of the Andes, they would have unknowingly taken this little insect hitchhiker with them, because they can't tell from the outside it's in there. And when they got to new geographic destinations, the weevil would have emerged to complete its life cycle. It would have been completely cut off from its former parental gene pool, and then over time, mutations occur in the population, and this could lead to species divergence given enough evolutionary generations. So by sequencing the different species of this insect complex, you can reconstruct their evolutionary history. So who's related to who, and at what are the timing of them diverging from each other. So the first thing I did was I worked with my collaborator, Jesus Alcazar, who is one of 5 people who is expert of the Andean potato weevil in the world. So I was really lucky to work with him, and we collected samples of the weevils from throughout the Andes. And because there's 10 of them, I've color-coded them to kind of keep track of them. So the warmer colors are more from the southern Peruvian Andes, and the cooler colors are more from the northern Peruvian Andes. And so after doing collection and DNA extraction as well as sequencing, I constructed a phylogenetic tree using 4 different commonly studied gene markers, so 2 mitochondrial and 2 nuclear. And I concatenated the sequence information from these genes in order to construct the phylogenetic tree that you see here. So there were a few major conclusions from this phylogenetic analysis, and again, I've color-coded the specimens from each species. So what I found is that the large majority of central and southern weevil species are more related to the last common ancestor of the complex. So it's likely that the ancestor of this entire insect complex also originated in the southern Peruvian Andes, right around where the potato was domesticated. There was also a high degree of relatedness between the central weevil species as well as the southern weevil species, which indicates that there was continuous migration with human populations and their potatoes, or continuous trade with potatoes in human populations. And what I found is that while these 2 northern weevil species are very geographically isolated from the rest of the complex, and they're highly related to each other, and the central and southern species are highly related to each other, that we have one outlier. So we have a weevil species that's geographically very close to the other southern weevil species, but actually more related to the northern weevil species. And when I used a molecular clock to estimate when the timing of these species diverging from each other would have occurred, what I found is that the central and southern weevil species are the oldest. So again, most likely the ancestor of this complex originated from around the Lake Titicaca Basin, and that the northern species diverged next. Our outlier weevil species is the most recently diverged weevil species. So what this could mean is that after a period of geographic isolation in the northern Peruvian Andes, people actually migrated back to the southern Peru with their potatoes and led to the establishment of this new outlier weevil species. And I did a few other population analyses of this outlier, and I found that it likely experienced some kind of population bottleneck after it was established in this southern Peruvian region. So what this could mean is that despite being very geographically close to these other human populations growing potatoes, there's some kind of cultural or ethnic barrier prohibiting people from trading their potatoes in this region, leading to this weevil species being very genetically distinct from the rest of that complex. But I was left with a lot of questions, which is often the case in research. What is the actual timing of this backwards southern Andean migration? Did it occur thousands of years ago? Did it occur in relation to Inca practices of moving different folks around? Not quite sure. And what are the time periods of these other migration histories and trade patterns in the Andes? And right now, for the molecular clock that I calibrated, I'm using generalized estimates of mitochondrial recombination rates. But like hominid ancestors, we really need a fossil reference in order to more precisely calibrate when these events would have occurred. So I'm actually moving toward doing some sedimentary eDNA collection. So what I have on the right side of this screen is an ArcGIS map of approximately where that northern site was and approximately where the southern site was. And this algorithm in ArcGIS generates a least cost path. So the route that people would have taken that had easier paths to walk on, right? They're not walking over water, they're walking in valleys. And I'm working with archaeologists to identify sites along this path where there's either known sites of potato domestication or perhaps some new sites that we can identify of potato remains, and taking soil samples to extract all the species available in the soil, and then isolating any potential archaic weevil DNA with the genomic database I've built up. And so again, I just wanted to highlight a few different projects that I've really built up on and not concluded, but made a good jumping off point during my time at UCLA. And I really think that working with Abby and being on campus here has really developed this thought process and this research program that I'm working in now of how does our evolutionary past affect populations today, and how can we relate that to their health, and how can we use that as anthropologists to inform local capacity building as well as conducting ethical community engaged research. And so I just want to say thank you to all of my collaborators at these different sites and our Quechua, Maya, and Sherpa participants, as well as all of the potato farmers that let us destroy their crops so we could get some insects, and my funding sources. And since Abby and Kim couldn't be here, but were a big part of my time at UCLA, I thought I'd throw up some pictures so they could be here in spirit. So thank you guys for listening.